All right, everybody. Thank you so much for being here at our first Science on Tap of the academic year. We're so excited to get this started again. These are monthly events, and so thank you for being here. This is a hybrid event, so there's folks that are also online, so thank you for those that are online. Hopefully you all are someplace with some good food and drink and in a comfortable chair, and you're joining us. For those folks that are here in the room, thanks for coming out to being at Hatfield. My name is Cinnamon Moffett. I am the program manager at Oregon State University's Hatfield Marine Science Center, located in Newport, Oregon. I will be your host for this evening. A couple logistics as we get started with things. For folks that are online, if you have any technical issues, we have a volunteer named Mike who is watching the chat. So feel free to use that and let us know if you're having any issues. Also, for those folks online, this is a webinar format, which means that your cameras, mics, and screen shares are disabled. But please, please, please ask us any questions that you might have using that chat function, and Mike will read them out to the folks here online at the end of today's presentation. For those folks that are in the room, if you haven't been with me before, we also will have you all ask questions at the end of today's presentation if you have them, but you will need a mic to do that. You can either raise your hand and I'll bring you the mic in my hand, or you can go to the mic stand right over there, but that means that folks that are online can hear your questions as well. Okay. Couple of other little announcements. I want to make a plug. I might not need to make a plug, but I'm going to make a plug for next Science on Tap on October 5th. We are going to do a shark dissection. And so it is going to be pretty cool. The Big Fish Lab is going to come and talk a little bit about sharks in Oregon. And then we're going to use this room and some GoPro cameras to get a bird's eye view of the shark dissection. For those folks in the room, you'll be able to broadcast it up on this big screen and you'll be able to see all the things that are happening. So if you're interested in that, that'll be October 5th. My guess is you'll want to come early. So that'll be a good one. So making a little plug there. Okay, but for the reason that we are all here, I also want to let folks know we are recording tonight's event. So if you're interested in sharing it with others or watching it again, you can go to HMSC's past seminar page. It'll be there in a couple of days. But give a little plug for who we've got with us tonight. We are excited to have Dr. Rene Boiteau here to talk to us about his work. But first, just a little bit of background. Rene is a chemical oceanographer and assistant professor with the College of Earth, Ocean, and Atmospheric Sciences at Oregon State University. He received his PhD in 2016 from the Massachusetts Institute of Technology and Woods Hole Oceanographic Institute. He then went on to do a postdoctoral fellow in the Pacific Northwest National Laboratory before coming to OSU in 2018. His research group investigates the natural and human-induced effects of metal on ocean and terrestrial ecosystems. We are so excited to have him here to speak to us about this topic that we've all been kind of thinking about for a couple years, and so we're excited to learn about where we're at. So I'm going to hand this off to Rene. Rene, the floor is yours.
Great, thanks a lot, Cinnamon. I'm Rene Boiteau, and my research interests really focus around how metals impact the environment. And today we're going to talk about iron fertilization. So what it is, why it's a big deal, and what we really know about its effects on the ocean. Right? And so like many aspects of science, usually when iron fertilization makes its way into the news and international conversation, it's due to some sort of controversy. And so about a decade ago, there was a controversy that hit particularly close to home here in Oregon. It was in July of 2012 when the fisheries off of Canada chartered a fishing boat to dump 100 tons of iron into the North Pacific Ocean. And so this project had the goal of fertilizing plankton, which had the intended consequences of increasing salmon populations and also sequestering carbon. Right, so this made international news because it was a really controversial project, right? The dumping of nutrients into the ocean violates international bans on geoengineering and ocean pollution, and the science that backed up this type of effort was really, really unsettled, right? So we can still ask questions of, did it work or did it have unintended consequences, right? And in order to understand that, I think we have to back up and think about what they were really trying to do. Okay, so iron fertilization or ocean fertilization more generally is the idea that you can dump nutrients into the surface ocean to increase the growth of phytoplankton, just like you would add nutrients, fertilizer to your garden to increase plant growth. Right, so if you go take a ship out to some regions of the ocean and add the nutrient that is limiting algal growth in that area, you will get increase in what we call primary production. And that algae are basically taking CO2 from surface waters and building biomass from that, right? So that CO2 that they've taken up gets replenished from CO2 dissolving into the seawater from the atmosphere, and has the effect of drawing down atmospheric CO2 concentrations. When the algae die, they're either eaten by higher trophic levels, and so there's the potential for that carbon to cascade up into fish and other populations. And then when larger things die, they sink, right? And so ultimately, this takes that carbon that was originally dissolved in the atmosphere, is taken up in the surface ocean, and gets transferred into the deeper layers of the ocean. Now, the ocean is a stratified system. That means that the surface layer is rarely in contact with the deeper layer. The deep waters of the ocean are isolated from the surface with a residence time of about thousands of years. So if you can take that carbon and transfer it below 1,000 meters or so in the ocean, it's effectively locked away from that surface atmosphere system, right? And so again, the goal of iron fertilization is to transfer carbon into higher trophic levels and ultimately export it into the deep ocean from the atmosphere. So why might iron be a good target for these fertilization efforts? Well, in order to understand that, we really have to go back to the beginning of the ocean. Literally. So, the Earth formed about 4.6 billion years ago. And at that time, it was molten rock and gases. And eventually, it cooled to the point where we could have liquid water. And in that early Archean Earth and early Archean ocean, the chemistry of seawater was dictated by these volcanic processes. So there was lots of iron in the ocean, lots of sulfide in the ocean. And these are the building blocks that the early forms of life that colonized the ocean had to deal with, right? So this is an artist's rendition of what that planet might have looked like. And you can see it looks a lot like the parks at Yellowstone, right? So these are places where in hydrothermal vents, bacteria can take up the sulfide that's produced from volcanic activity, and they use that to actually photosynthesize fixed carbon and build biomass. And we think that those are basically the ancestors of things that lived in the early ocean. So life evolved in this ocean that was filled with iron. And organisms discovered early on that iron was a really wonderful element to use to shuttle energy between molecules. Right? So a lot of the enzymes and proteins that organisms developed to build biomass and carry out essential functions rely on iron to transfer energy, right? And so organisms on the planet today have this legacy. They use iron for many, many essential processes: DNA replication, photosynthesis, respiration, that's the conversion of carbon into energy, nitrogen fixation, the conversion of nitrogen from gas form to a useful form of life. They deal with stress with iron-containing enzymes, right? And the list goes on and on and on, right? Iron was very abundant. It was a great catalytic center that allowed organisms to carry out different reactions that are essential to life. And this worked great for many, many years, for a billion years. Eventually, something screwed everything up, and that was the development of oxygenic photosynthesis. And so oxygenic photosynthesis is the type of photosynthesis that we're mostly familiar with today. That's what plants on land use. That's what most of the algae in the ocean use. And that's taking sunlight and CO2 and water and converting it into organic matter that is used to build cells and oxygen. So oxygenic photosynthesis showed up in the ocean about 2.7 billion years ago. And two things do not mix oxygen and iron in water, right? So when you have oxygen and iron around, they form rust. And that is very, very insoluble and just crashes out of solution. So when oxygenic photosynthesis arise, this is a period of Earth's history we refer to as the Great Oxidation Event. And one of the hallmark characteristics of rock formations, marine sediments that are scattered around the planet that originate from this time are these what we call banded iron formations. And these are relics of this process of the ocean rusting out. So you can see these beautiful orange bands that each represent, a pulse of oxygen produced by algae that basically converted a bunch of iron in the ocean into rust that settled out of the ocean, right? And so over the course of the Great Oxidation Event, we went from a iron-rich ocean where you had all of the iron you needed for biological processes to an ocean where iron is very, very, very poorly soluble. And all of a sudden, organisms have this issue to deal with. Now, this essential metal is only available in places where you're really close to some source. It took oceanographers a really long time to recognize that iron was a really important nutrient in the ocean. It actually wasn't until the late 1980s. And the key to understanding that is certain regions of the ocean we refer to as high nitrate, low chlorophyll regions. Right, so chemical oceanographers like myself measure distributions of chemicals across the ocean to understand the processes that are going on. So in the case of biological processes, the most important thing to understand is the supply of nutrients. And in the early days of chemical oceanography, the focus was really on nitrate. Nitrogen is the nutrient that is required at the highest concentrations. And so it was basically thought that algae would grow until they ran out of nitrate, and that would cap productivity. But when we started to map out nitrate across the ocean, it became clear that something else was going on. And so this is showing here a plot of sea surface nitrate. So you can see warm colors indicating high concentrations in red and cool colors indicating low concentrations in blue and purple. And so most of the ocean has very low nitrate because it's been all stripped out by organisms that need it to grow. But about a third of the surface ocean has pretty high concentrations of nitrate that persist. And this was a puzzle for a really long time. It turns out it was really hard in those early days of oceanography to understand how much iron was really in the ocean. And this video gives you sort of a sense why, right? So this is one of the preferred methods of collecting seawater for oceanographers. This is a clip of showing some students collecting samples aboard the Oceanus, the retired OSU vessel. And you can see that the instrument they're deploying is called a CTD rosette. So it has an instrument package on the bottom and all these bottles on the top, and then this conductive wire that goes to the surface. And so we can lower this down into the depths of the ocean, measure the chemistry of the ocean as it goes down. And then as we pull it back up, we can trigger the bottles to close through that conductive wire and collect discrete seawater samples. So we're collecting this seawater using a system that has a steel frame. Inside of each of those bottles are steel springs. The wire is steel, and we're on a steel ship. All of that is loaded with iron, and the resulting sample that you get has a lot of iron contamination, far, far greater than the concentration of iron in the seawater itself. So in order to actually study trace metals like iron in the ocean, we had to develop completely new techniques, and that was very difficult. Fortunately, there were a handful of brave scientists in the 80s who developed trace metal clean oceanography, trace metal clean techniques. And so they developed new systems for sampling seawater that made use of plastics and other materials that didn't contain metals that would contaminate. They developed ways to process samples cleanly on a ship that's made of iron. This is a couple of photos from my colleague, Kristen Buck, showing two different examples of makeshift clean rooms that we can set up on a ship. So the one to the left is showing a bubble where we basically coat a lab in plastic and then have these HEPA filters that provide clean air. And then the one on the right is showing a van that we can actually, it's basically a modular clean room that we can load onto a ship and do all our sampling there. And you can see some trace smell clean bottles that are suitable for iron sampling. And so it was a huge effort to first develop these tools that are very different than the typical tools that oceanographers prefer. Oceanographers love stainless steel. But when we did that, we started to be able to measure iron concentrations and realize that they're very, very, very low, especially in these high nitrate, low chlorophyll regions. And so in the late 1980s, oceanographers started to recognize, okay, in these regions, iron concentrations are really low. Maybe that is the limiting nutrient. And John Martin and colleagues proposed some of the first iron fertilization experiments to test the hypothesis that iron was the limiting nutrient in these regions of the ocean. So they traveled in a ship to the North Pacific, one of these high nitrate regions of the ocean, collected seawater with these trace metal clean techniques. So they didn't contaminate with iron. And then they were able to incubate that seawater with different amounts of added iron. So they're able to very carefully control how much iron was going into their bottles. And they measured over time the concentration of chlorophyll, which is chlorophyll is the pigment used by algae for photosynthesis. So it's a proxy for algae abundance. And they measured the concentration of nitrate. And in this first paper, they found that when they didn't add any iron, they saw no plankton growth and no nitrate drawdown, or very, very little. There's probably still some contamination in these experiments, these first experiments. And as they added more and more iron, they got more and more phytoplankton growth, and they were able to see complete nitrate drawdown. So this was a really big deal because this was the first demonstration that, if you add iron to these waters, you can actually get phytoplankton to draw down that excess nitrate. And ultimately, it's probably iron concentrations that are governing the productivity of the ocean in parts of in the high nitrate, low chlorophyll waters of the world. And again, these account for about a third of the surface ocean. So it's a large area. So John Martin had a bit of a flair for the dramatic. And in presenting some of these results at one of the early conferences, he stood up there and said, give me a half a tanker of iron and I will give you an ice age. And this is basically a recognition that if you imagine taking iron and adding it to all these regions where you have those warm colors, you would create a phytoplankton bloom that would take down all that nitrate along with carbon. And when the algae died, that carbon would sink into the deep ocean, and it would have huge impacts on atmospheric CO2 concentrations. So this was really a major paradigm shift in oceanography, in chemical oceanography, because at the time, it was the first recognition that a micronutrient, a metal like iron, had the potential to control ocean productivity on a global scale. Right? There was a lot of enthusiasm about this result. It meant that you could actually envision geoengineering strategies where you go to places in the planet, add iron, and use that as a strategy to sequester CO2 or boost fishery stocks. And of course, that was also met with an appropriate level of skepticism, saying, what could possibly go wrong if we start, these massive scale manipulations, right? Regardless of which side you fall on or fell on at this time, it was very clear that we needed to understand iron fertilization, this process in the ocean, right? And really pin down whether iron was really limiting in all of these different regions of the ocean where you have high nitrate concentrations, right? Is this really a global control on productivity? And so the next decade really was followed by a number of large-scale iron fertilization experiments where scientists went out on ships, and these projects were arranged by countries all over the world that coordinated and conducted these types of experiments. They dumped iron sulfate into the surface waters where you had excess nitrate concentrations and monitored what happened over time, right? And ships can go out for maybe a month or so. And so this is sort of the time scale of those experiments, right? And the goal was really to figure out, across these different places in the planet where you have high nitrate concentrations, is iron the limiting nutrient that caps productivity? And if you add more iron, do you get an increase in productivity? And does that ultimately result in a change in the ecosystem and an increase in the export of carbon to the deep ocean? Right. So after a dozen or so of these experiments, researchers got together and compared results, and the results were complicated. So one result was really, really clear and really, really important. So in all of these experiments, there was an increase in primary production when iron was added to these high nitrate, low chlorophyll regions. Right? And so this is a table that came out, a table from the same paper, Boyd et al., that summarized the results. And basically across the board, we had increases in primary productivity that were measured within fertilized patches compared to outside the fertilized patches. Really nice demonstration that, yes, iron is really the limiting nutrient in these broad regions across the ocean. What wasn't consistent was the actual types of organisms that grew, either the types of phytoplankton that grew or the growth of zooplankton that are eating the phytoplankton. And what also wasn't consistent was the effect on carbon export, right? So you can see in red, most of the iron fertilization experiments that were conducted didn't result in a significant increase in the flux of carbon from the surface to the deep ocean, but some of them did, right? So looking at all of this, there was just a really unclear picture of what the effect, the main long-term effects of iron fertilization would be. Right. And I think so oceanographers reflected and said, what do we really know about the iron cycle in general? And the answer is not much. So this is a plot here showing the locations where dissolved iron measurements had been made as of 2003, sort of at the end of this decade of iron fertilization experiments. And that map looks pretty sparse. So we didn't really know the answer to zero order questions about iron in the ocean. Like, how much iron is in the ocean? What are its major sources and sinks, right? And so there was a recognition that we had to really just figure out the basics of iron in the ocean in order to understand the variable impacts of iron fertilization. Right. So the group of scientists got together, formed an international group called Geotraces, and this is really one of the most exciting projects I think that has been going on in oceanography today. Over the past decade, this has been a concerted effort to map out the distribution of iron and other trace metals across the entire ocean and really figure out where it's coming from and where it's going. So we're at the tail end of Geotraces now. So this was conceived in the mid 2000s, started late 2000s, and we're about a decade into Geotraces. And this is showing here a 3D map of the intermediate data product that was released just last year in 2021. And this is our first view of iron distributions throughout the Pacific Ocean. I just love this picture. So you can see that, again, warm colors are indicating high concentrations red, cool colors are indicating low concentrations in blue. And really for the first time, we can see that there are these pulses of iron that are coming from hydrothermal vent activity along ocean ridges, supplying iron to the ocean's interior. We can see iron coming off of continental margins from dust and from sediments. And throughout the surface ocean, we can see really, really low concentrations of dissolved iron, where organisms are taking up that iron and packaging it into their cellular proteins. The other thing that's really notable is that no matter where you are in the iron, dissolved iron concentrations are very, very, very low. So these concentrations represent up to 1 or 2 times 10 to the minus 9th moles per liter, very, very low concentrations. And so the outcome of Geotraces has been a new view on the processes that are supplying iron to the ocean, how much iron is in the ocean, and how long it lasts there. And so what we've started to discover over the past decade is that the main sources of iron to the ocean are atmospheric deposition, particles from the atmosphere depositing in the surface ocean, volcanic activity, like at hydrothermal vents in mid-ocean ridges, and then continental margins where we have fresh water coming in and sediments that release iron. And then there's a whole process of internal cycling that's also very active. And so the takeaway of all of this is that iron concentrations are really, really low. And in the surface ocean, iron lifetime is really, really short. Iron spends only a year, really less than a year in the surface ocean, which is very different than many nutrients. And so the crux of this is that inputs of iron are really important. They are very dynamic. Things like atmospheric deposition and continental margins are basically governed by atmospheric circulation and ocean circulation. These things can change quickly, and those can really rapidly change the global iron cycle. So iron is an essential micronutrient that is governing the amount of productivity in vast swaths of the ocean, and it has the potential to change really quickly, on the timescales of human lifetimes. So we really have to understand iron. Okay, so at the same time, there was an interest in understanding the impact of variable inputs of iron into the ocean, right? And so iron fertilization is a natural process in many cases, right? There are changes in the sources of iron that can fertilize regions of the ocean that didn't have enough iron at one point in time. In the context of paleoceanography, the study of Earth's history, it has become really clear that iron is a likely modulator of global climate on timescales of the past millions of years. So this slide is highlighting a couple of studies that point out that there's a lot of evidence that during the last ice age, about 20,000 years ago, when temperatures were much colder, CO2 concentrations in the atmosphere were much lower, iron fertilization was a likely driver of carbon sequestration of the ocean. So there's really good, so if you look at sediment cores, there's a really strong correlation between atmospheric CO2 drawdown and the amount of iron being deposited in the Southern Ocean. And the Southern Ocean is today is the largest iron limited ecosystem on Earth, right? So in the past, there was a lot more iron being delivered to the Southern Ocean from South America than there is today. It's very likely that that resulted in more algal growth in this part of the ocean, lower CO2 concentrations, because that is basically pumping carbon into the deep ocean. Dust is a really important source of iron to the ocean, and the effects are not always things that we would consider good, like sequestering CO2. So there have also been noticed some very strong correlations between the delivery of dust from the Saharan desert and red tide events in the Gulf of Mexico. So the Gulf of Mexico cares a lot about red tide because this really shuts down commerce in that area, and they're fairly frequent. And there's pretty good timing between events that blow dust all the way from the Saharan desert across the Atlantic and into the Gulf of Mexico. So this is one of the early papers that made that connection. And the idea here is that when dust comes into the Gulf of Mexico, it promotes the growth of trichodesmium. It's a type of algae that also takes nitrogen from the atmosphere and turns it into an organic form. And it seems that the organic form of nitrogen that's created by the trichodesmium is very tasty to the red tide organism that causes these harmful algal blooms. So you get this ecological succession that results in hazardous conditions for aquaculture and for humans. There are other types of iron, other sources of iron that can fertilize the ocean. So this is a result from a student at Oregon State University who graduated last year from the Marine Resource Management program, Leah Adroli. So she was advised by myself and Ricardo Letelier. And she was studying these anomalous blooms of algae that occurred in the North Pacific Ocean, just off of Hawaii. And one of the things that she noticed is that the strongest bloom events coincided with years when the Hawaiian volcano erupted. So this is an image that she put together of chlorophyll fluorescence monitored by satellite. So this is, a picture of the fluorescence of the surface ocean taken from space. We have these resolved over time across most of the ocean. So this is from July 2018. We have this bloom, and in other years, this bloom didn't occur. And when you look at that in the context of many different things, the thing that really stands out is aerosol optical depth. And this is a measurement of how many particles are absorbing light in the atmosphere. So again, this is an image that's taken from a satellite. And so you can see that aerosol optical depth is very high in this region. Again, that didn't occur in other years. And tracking where these particles came from, it's very clear that they came from this eruption that occurred in June from the Kilauea volcano. Right? And so there were a lot of other analyses that are part of this study, but the fundamental explanation for these blooms was that the volcano released a bunch of ash that contains a lot of iron. They were deposited in this part of the ocean where other nutrients were provided by upwelling from deep in the ocean. And these two factors combined give you these really massive blooms that only occur in years where we have these eruptions. Iron's important. This isn't the only place where volcanoes seem to fertilize the ocean with iron. And so we're about to come full circle. And so in 2010, there was noticed an interesting correlation. So there was a volcano that a volcanic eruption in the Aleutian Islands off of Alaska in 2018. And similarly to the Kilauea volcano, there's evidence that this volcanic eruption created a phytoplankton bloom. So the Aleutian Islands are in the North Pacific, and the ash spread into one of these high nitrate, low chlorophyll regions. So this picture here is showing satellite image of where that volcanic ash was deposited. So here are the islands, and the ash was spread all around this region. Well, in that later that month, if you look at chlorophyll fluorescence, so basically if you look at algal growth, along that path of ash, you have very, very high algal abundance. And if you compare that to other years, this is just looking at the previous year, that didn't exist, right? So this is evidence that that ash is a likely contributor to productivity, algal growth in the North Pacific at this time. 2 years later, it was observed that the number of sockeye salmon in the Fraser River had one of their best years ever. And it was proposed that maybe there's a connection, right? The sockeye salmon were young in this region 2 years before. Perhaps this contributed to the run. I'll note that if you look at other salmon populations, they don't show the same increase. And there's a lot of speculation that goes into that connection. It's an untested hypothesis. But that was basically the basis for that controversy that occurred in 2012 that I had mentioned before. And so, the thought of iron fertilization in the North Pacific affecting fisheries led to these projects to fertilize the ocean with iron and boost fisheries. It didn't just happen in Canada, by the way. There have been a number of other incidences where they're trying to do the same thing. There was another case in Chile in 2017, and there's interest in these types of things. So I would say that the controversy around these is really that these projects are taking what's really an untested hypothesis and using that to make management decisions on how you're going to change ocean ecosystems. And the science really isn't in. And we can talk a lot about the issues with that. But for now, let's talk about just did it work? Is there any evidence that this has the desired effects? So let's back up and remind ourselves what those effects were intended to be. So the first one was that they were intended to fertilize plankton. And we can assess that in some ways by trying to look at satellite data around the region where the iron fertilization occurred, right? So again, in 2012 in July, this is end of July is when the iron fertilization occurred. And again, this was not a scientific experiment, so we don't have a lot of details that we would like to have to really evaluate these things, but you can see that, you know, the next month, there's a large phytoplankton bloom, roughly in the area where the fertilization occurred, right? So it probably did affect productivity. It probably did create an algal bloom. That's consistent with the science, right? In all of these iron fertilization experiments, there was a bloom. Did it boost salmon populations? Well, it's really hard to assess that. You know, I mean, this assessment is kind of like taking, you're trying to validate a drug by, you know, a new drug by giving it to one patient, you know, seeing if they get better, and then trying to decide, well, did the drug make them get better, or did they eventually just get better, right? So you can track salmon populations that followed, you know, some went up, some went down. Basically, we have no way of figuring out whether this had any effect or not. Did it sequester carbon? Again, we have no way of evaluating that. This wasn't done in a way where we could actually assess what was going on. So, you know, the takeaway, I think, the lessons learned from these types of projects is that, you know, as a management practice right now, there's no way to really evaluate its effectiveness. We don't know the long-term consequences of that, but it is really important to understand what those consequences are, because this is a process that happens naturally, and, you know, there is this interest in exploring geoengineering strategies, right? So it's worth sort of taking a step back and thinking about, what do we really know about iron fertilization, and what do we not know and need to understand? So the known effects. So it's well established at this point that in about a third of the surface ocean, the base of the food chain, growth of the base of the food chain, is limited by the availability of iron, right? And so if there's a natural or a human fertilization of these regions with iron, it leads to an increase in primary production. In some cases, that fertilization transfers carbon to the deep ocean, right? In some cases, it doesn't, and we don't really understand what governs outcome A versus B. And then we know that some of the major natural sources of iron are dust and sediments and volcanic activity, although we're still trying to get a better handle on what their relative contributions are and how variable they are through time. And that's basically what we know about iron fertilization. And what we don't know are the more long-term effects largely, right? So in some cases, maybe iron fertilization could sequester CO2. It could also perhaps increase fishery stocks. On the flip side, there's some evidence that iron fertilization can lead to an increase in harmful algal blooms that would have an adverse effect on fisheries and obviously pose risks to human health. And then there are some lines of evidence that in some cases, iron fertilization could lead to expanded oxygen deficiency regions, and also lead to the production of other greenhouse gases that are very potent, like nitrous oxide, right? All of these are some of the more long-term effects associated with iron fertilization. And we just haven't done studies to track what happens to an ecosystem that's fertilized with iron over time. Okay, so what is next, and where do we go from here as a field? So, you know, we spent basically a decade in the 2000s trying to really pin down the effect of iron fertilization with these experiments. Then we kind of took a step back and said, well, let's just figure out the global iron cycle. How much iron is in the ocean? Where's it coming from? And we're starting to get that pretty well pinned down. And so there's, I think a lot of people in the scientific community are kind of coming back to this idea of iron fertilization as a potential remediation strategy, or just as a scientific tool to understand the impact of iron fertilization. So there was a recent report from the National Academy of Sciences, which is one of the organizations that helps decide science directions across the country. And they were looking at, you know, what are some potential options to deal with the fact that CO2 concentrations are really out of control, rising in the atmosphere, and what could we possibly do about it? And the bottom line is that, you know, the conclusion of this report is that iron fertilization is one of the best options to explore as a possible carbon dioxide removal approach. So, I think we have very different tools that we had back in the 90s and early 2000s to study the long-term effects of iron fertilization on marine ecosystems. And I think there's growing interest in using them to actually study experiments where we fertilize controlled regions and track what happens long-term. There's also an upcoming project that's part of Geotraces to understand one of the most important natural iron fertilization processes on the planet, and that's in the Southern Ocean. So I mentioned earlier that the Southern Ocean is the largest iron limited system on Earth, largest iron limited ecosystem on Earth. So changes in iron dynamics there have really big consequences for global ocean ecosystems and atmospheric CO2. Well, it's also one of the places where conditions are changing the most rapidly due to changing temperatures, right? So in Antarctica, ice melt events are becoming more and more rapid, and that's having cascade effects. And one of the biggest effects that increased ice melt can have on the ocean ecosystem is how much iron is delivered to those surface waters, right? Because ultimately, it's the amount of iron that gets there that will determine how much productivity there is. Well, when you get seasonal melt events around the Antarctic margin, they coincide with some of the most productive waters that you see in the world. So this is an image taken during the spring melt in the Amundsen Sea. And so much algae grows in that time period that it looks like green Kool-Aid. And somehow this is driven by pulses of iron that are mixing into those surface waters that already have lots of nitrate and phosphate. And so there are two upcoming research expeditions that are part of this Geotraces program that are investigating where is this iron, what are the sources of iron to the Amundsen Sea, and how will those change in response to future changes in climate scenarios. So our group at Oregon State University is really excited to be a part of these expeditions. The first is leaving later this year, and the next will be 1 year later, to really understand this process. So hopefully in a couple of years, we'll have a better sense of how Southern Ocean ecosystem dynamics are going to change. Okay, so to summarize, ocean iron fertilization is a process that occurs naturally and has a huge effect on ocean productivity and carbon cycling. And because iron has such a short lifetime in the ocean, the sources of iron that are going into the ocean have the potential to change on human timescales. And so it's really important to really pin down what those changes are going to look like, because they'll govern the future of ocean ecosystems and carbon uptake. So there is really a strong need to better understand what factors ultimately govern the response of the ocean to iron fertilization, and what are some of the long-term impacts, especially with regards to fisheries and for carbon sequestration. And with that, I'd like to thank my research team. So Leah Droli and Ricardo Letelier were instrumental in that volcanic dust study. The rest of my research group, Nicole Coffey, Peter Bright, Maria Alvarez, Kate Kuba, and Peter Chase, are helping work on some of these projects looking at iron dynamics in the ocean. And then many great collaborators here and elsewhere: Kristen Buck, Jim Moffitt, Claire Reimers, Dan Rapetta, and his students Jay Lee and Yulia Madalena Stringa. And of course, funding from the National Science Foundation and the Simons Foundation. And thanks for listening. I'd love to take some questions.
That was great. Thank you so very much. I just want to pause and encourage those folks that are listening online to go ahead and put any questions into the chat. And I'm looking at Mike to see if there's any there yet. And he says no. So that means that we can open it up to questions in the room. Looks like Megan is going that direction. So Megan, I'm going to meet you there so I can turn that one on.
Testing. All right. Rene, that was awesome. I learned so much. I have a lot of questions, but I'm going to limit it to 2 for
Awesome.
now. My first question is there some kind of ocean water that would be more vulnerable to like a bad algal bloom versus like sinking all of the like die off and sinking all of the plankton to the bottom? Like,
Yeah, that's a really great question. So the ultimately the question is what governs the community that establishes after iron fertilization, and what effect does that have? So if you have a bloom of diatoms, these are algae that have silica shells that are very heavy that will sink out of the water column. So so the short answer is we don't quite know, but it has to do with the types of organisms that are already there and the other nutrients that are there. And these are probably linked together. So ultimately, what we should understand is, how does the chemistry of the waters that's fertilized affect the outcomes? So one possibility might be if there's a lot of silica in the water, then you can have diatoms that form heavy shells that will ballast them and increase the sinking, right? If you don't have the silica, then you might not have that effect. That's a possibility. Again, this is untested hypotheses. In the case of red tide, it seems to be linked to the bloom of these organisms that are fixing nitrogen. And those organisms do well in places where there's very low nitrate to begin with. So the Gulf of Mexico has very, very low nitrogen concentrations. These trichodesmium organisms are fairly abundant there because they can take nitrogen from the atmosphere and grow. And so in places like that, you might have the community set up to respond to iron deposition to create the conditions that would lead to harmful algal blooms. But again, these connections really need process studies that try to track the transitions, and we don't have that right now.
yeah, thank you. And my other question is that really cool rotating map that you showed, where that had the transects through the ocean.
Yes.
It looks like there was a lot of iron off of the coast of Japan, maybe. I think it was maybe that
We froze.
that one. Yeah. Is that Japan up there? On top right now.
Yeah. Mm-hmm.
Yeah. So why is there so much iron there?
That's a good question. I would have to go back and read a bit more, but in general, a lot of the iron that ends up in the Pacific Ocean is coming from sediments. So in sediments where you have a lot of organic matter accumulate, that organic matter breaks down first by using up all the oxygen in sediments, and then starts breaking down through microbes that they have to respire. So if oxygen runs out, they can't respire with oxygen. They end up basically dumping electrons onto iron minerals and releasing that as iron 2, which is that form of iron that existed in the Archean Ocean. And these sediments that have a lot of organic matter can provide iron to overlying waters. And if that iron is in the right form, it can be perhaps transported quite far distances.
Okay.
So a lot of those regions where you're seeing offshore transport of iron, it's iron that was present in sediments. There's also a lot of dust that sometimes comes from Asia across the Pacific Ocean. And that's another potential source, but it depends on timing and things like that. So
Cool. Thank you.
Yeah.
All right, I'm going to pause. Any questions online? All right, any other questions in the room?
So I'm old, and so is my science. Specifically, this idea that I'm about to ask you about comes from a 1970s copyright basic oceanography book that I did a correspondence course in 1989. With that said, what that book proposed is that it was the iron fixation that came with oxygen production of early cyanobacteria and anaerobic photosynthesizers that eventually overcame the iron burden that was preventing oxygen in the atmosphere and enabled a green earth and most life that requires oxygen. With that said, and with my brief synopsis of that, what is your take on what enriching the ocean environment, what effect would that have on available oxygen for those of us for whom that's important?
Yeah, that's a really fascinating question. So I think the scales are really different. So I think that's absolutely correct. Basically, in the early Archean ocean, there was a tremendous, we don't really know how much, but there was a lot based on these banded iron formations of iron in the ocean, right? And in similar anaerobic settings, you find 1,000, 10,000 times more iron, dissolved iron, in those types of waters compared to what we see in the ocean today. So you imagine the ocean is filled with that 10,000 times more abundant iron. That's a huge buffer against oxygen that's being produced. And so the great oxidation event is basically these oxygen producing bacteria first eating away all of that iron that's in the ocean, which is a huge amount, creating these banded iron formations. And only then can you have the accumulation of oxygen in the atmosphere. And so it obviously took a really long time to get to the 20% of oxygen that we have in the atmosphere today. So when we're talking about iron fertilization of the ocean, the magnitude is much, much lower. The only, I didn't get into this in this talk, but when you add iron sulfate to seawater, most of it just turns into rust. And this is something that is actually not so easy to track. But basically, you're not creating a huge amount of iron that would react with oxygen. And so it would really have negligible effect of even just dissolved oxygen concentrations. But it's a really, really good question. So fortunately, we wouldn't all suffocate. Yeah.
All right.
Or it would be very, very hard to get that much iron into the ocean.
We got another question coming to the mic. Hang on.
Okay. Hey, Kip Sherman, Oregon State University. So I'm curious about this map too, and why there's so few observations of iron in the ocean. Are there no in situ sensors to measure iron in the ocean? And if so, why?
Oh, yeah. So there are no in situ sensors for iron in the ocean. And the reason is because twofold. I mean, the main reason is that iron concentrations are very, very, very low. And so you need to make really, really sensitive measurements. Most of the types of things that we can analyze, even nutrients, sensors for those are looking at like micromolar concentrations. And here we're dealing with nanomolar concentrations. So the analytical tools that we have to measure those very small concentrations are not well suited to deployment at this point. The other big issue is contamination. So, and they kind of go hand in hand, right? So you also need to have very, very low concentrations of contamination. And a lot of the types of tools that we use, this is getting back to the same issue of, oceanography loves stainless steel, and trace metal analyses have issues with that. But I would say that, even if you were able to develop a trace metal clean platform, the sensor development is still a challenge.
Thanks.
Yeah, not impossible, I bet, but hard, very hard.
All right, I'm looking at Mike again. Anything coming in online, Mike? All right, for those folks online, please put your questions into the chat and we'll get to those. Otherwise, we're going to stay with folks in the room. Go ahead, Francis.
Hey, Rene, thank you for that. Super stimulating. I was curious about a lot of things, but the fact that you related that iron has a very short lifetime in the surface ocean, that really intrigued me, because in ecosystem ecology, we think about which nutrient ought to have a very, very long residence time in ecosystem, and those tend to be ones that are really limiting. So why is it that iron bucks the trend? It's a highly limiting nutrient, yet it has a very rapid, very short lifetime in an ecosystem. why is it hard for biology to conserve that and keep in the closed cycle? Or is it that you're kind of measuring the, you're just looking at iron 3 when it's really the iron 2 that you should be looking at for the turnover rates?
Yeah. So I think it gets down to like concentrations are really, really low. And when we think about these residence times, there's also a question about speciation form. And this is something that my research group really tries to understand. So not all forms of iron can necessarily be taken up by organisms. So there's a little bit of a disconnect there. But basically, the problem is that iron has such a low solubility that the concentrations of iron drop off too quickly away from sources. And the sources are all at these margins. And so vast swaths of the ocean just get such small iron inputs that there isn't enough in the water to maintain.
But if you have a small pool and small input, then you'd have a very relatively long residence time. It sounds like you have to have a small pool, but relatively high input to drive that rapid, that very short residence time in the system.
Yeah, we can chat more about the residence time calculations and everything, but yeah, the bottom line is the pool is very, very low. The inputs are still fairly low. Yeah, but the pools are so small that, yeah.
So I would propose part of the answer to, I think it's Francis' question, could be that the enzymes in so many of basic life processes originated in a very, very iron-rich environment. And so there was no reason for a conservation of that resource to ever become part of our essence.
Yeah, to some extent. I think, if you look at things like stoichiometries of nutrients in the ocean, right? So nitrogen and phosphorus have fairly conserved ratios relative to each other because they recycle in forms that are basically soluble. In the case of iron, right? If biology kind of buffers this biotic pool, and then it turns into this inorganic pool. In the case of iron, right? Organisms buffer this biotic pool that's bound up in cells, right? And when they die, the biotic pool, that biotic pool turns into inorganic iron that is insoluble. So a fraction of that gets stripped out. The nitrogen and phosphorus can be recycled, but the iron some of it becomes rust and gets removed, right? So if you look at like the turnover of nutrients in the surface community, and I think this kind of relates to what you're saying, you basically have a lost term of iron that you don't have for some of these other nutrients if you're out in the open ocean.
Any other questions?
Now we're getting to
online or in
chemical
the
photography.
room?
Love it.
All right, I'm going to selfishly ask one of my own. So we talked a little bit about the chemistry of the water that's receiving the input. I'm also curious about the timing of when that input would be added based on the speciation complex that might be available. Can you talk a little bit about that?
Yeah, so I guess, are you referring to like timing in terms of like, if you tried to fertilize a place in the ocean,
Seasonality.
different seasons,
Yeah.
would you have different outcomes, right? And I think that the answer is almost certainly yes. Yeah, and it's tied to everything else, right? So the nutrient and the present community will be different at different times of year, and something about the nutrients that are there and the community that's there is what should ultimately affect the outcome of an iron fertilization event. At least that's what our experiments so far suggest. The connections themselves are not well established. So, what the outcomes of those differences would be, we don't know.
Interesting. All right. We have run up against the end of our time block. And I know Rene is traveling tomorrow, so I don't want to keep him extra, but if we can just give our presenter a thank you one more time. And I think if you have any follow-up questions, you can reach out to Rene directly and you can ask those questions. And for me, I just want to invite you all again next month to come back and join us for the next Science on Tap. So for everybody in the room, thank you so much. For everybody online, I hope to see you again soon. And Renee, thank you so much. All right, everyone.
Thank you.
Until next time.
Yeah.
That's in ecology. We think about like, why this is
Yeah, I guess
limitation persists. It's uncontrollable losses, right? So that because we think that biology is going to hold on. But iron is one of these things where there's because of the F3, there's an uncontrollable losses out of the system. Because it ought to be closed, but it becomes a very open system despite this limitation. That could be I don't know.
yeah, maybe I'm thinking of residence time differently than you, because I'm thinking of like, I would expect kind of the opposite, where like shorter residence time would be more limiting, right? Like calcium has a super long residence time, so it's everywhere, so there's enough of it.
Well, I guess
So when I say residence time, it's really like, like how much time it spends in the upper 100 meters of the
I think like, let's say a nitrogen comes in, it's limiting, and biology uptakes, it's realized, and it's uptake again real rapidly.
yeah. So
It spins
Right.
at that surface layer, so it stays around.
yeah. So you're talking about a residence time of like how long does it spend outside of a cell versus inside of the cell? Yeah,
Just in the surface.
yeah, yeah.
Because like,
Okay, everybody online, I'm going to go ahead and stop the recording.
Right.
Thanks so much for joining us.
Yeah, I see what you're saying.
Every single nitrogen or phosphorus molecule out before it drops. And you would think that iron would be the same way, but maybe iron is different because it does have that Fe3 and
Yeah,
it's like, hey, I didn't see that.
right. I mean, it's oh, thanks. It's basically, yeah, that problem that biology tries really hard to keep the iron, and it's just a tough problem. Yeah. So a lot of the work that I do is trying to understand how microbes even do that, right? And so what we're just